I'm Maddie Broom,
And I'm Lexi Duscher.
and we are graduate students with the University of Florida working out of the Space Life Sciences Lab, which is right next to Kennedy's Space Center.
And we're most interested on trying to figure out how to keep astronauts healthy during long-term space travel.
Astronauts will continue to go to space and for longer periods of time, but how do we make sure that they stay healthy during space travel? Research on astronauts has found that their immune systems are highly dysregulated in space, and this may be in part due to the disruption of their normal, healthy microbiome, or the microorganisms that are associated with us, and this may lead to adverse health effects.
However, human microbiomes consist of thousands of different bacteria, and these complex systems are especially difficult to study in a space environment. Our lab focuses on using a simplified model system between one animal host, the Hawaiian bobtail squid, and its bioluminescent bacterium, Vibrio fischeri, to determine how a space-like environment influences the immune system in a host microbe association.
These squid have already flown into space on the Endeavour and Atlantis space shuttles, which further proves that they're great candidates for future spaceflight experiments. We plan on sending these squid into space again, but before we do so, we need to first conduct ground-based studies to ensure that the next time we do, it's the absolute best experiment possible.
To simulate a space-like environment in our lab, we place squid into these rotating bioreactors, and they simulate a microgravity condition, or near weightlessness, that is experienced by astronauts in space. The benefit of this system is that we can place squid in these bioreactors with or without their bacteria.
After the completion of the microgravity experiments, we are going to look at the genetic expression, or activity, of the innate immune response in the Hawaiian bobtail squid. The innate immune system is the first line of defense against invading pathogens and is found in all animals, making this work applicable to understanding how humans' immune systems might respond to their beneficial bacteria in space.
Our lab has already conducted a broad genetic experiment with the squid and their bacteria in the rotating bioreactors, and we have citations available on our page from these studies. But now we want to target specific immune pathways that may be altered in space.
With our current funding, we have already completed all the microgravity experiments and have already identified the target immune system genes that we're interested in looking at.
And now, with your help, we're hoping to complete our project. We next want to sequence the genetic material that we obtained from our microgravity experiments using a new technique that we're really excited about called nanostring. If you have any questions about the nanostring sequencing technology or about our project in general, please do not hesitate to ask, and we'll be happy to talk to you.
In the meantime, we really appreciate your help. No amount is too small, and every donation gets us that much closer to putting our squid back in space.